Hello everyone, welcome to the MedTech stage at the Biotech Future Forum. I'm Elena, your virtual host. Today's panel, Curing Cancer with Innovation, will explore breakthroughs in cancer treatment using cutting-edge biotechnology. Please welcome our esteemed moderator, Mr. Nenad Paunović. The stage is yours.
Thank you, Elena. Good morning, everyone. The name of this panel is Curing Cancer with Innovation, and this morning with the speech of esteemed Dr. Yankopoulos, we've heard about innovation in antibody therapies. Today we are going to speak in this panel on different kinds of innovation and different kinds of hot topic in curing cancer. Thank you all for coming, and I see we have a full room, and I'm particularly happy about that. And this panel is very specific because it has a moderator that is not from the industry. My background is in management, so each and every one of those people know immensely more than myself. But I believe it's also good because I will try to make them speak in the language that the rest of us can understand. And this is my main role here, is to find out what we are talking actually about. This panel is supported by the company Swiss Rockets with Serbian pilots. And I'm going to ask our panelists in the first round to just introduce themselves. Don't be shy. Share with the public how great and fantastic you are. If you do not, I will jump in. So it's better for you to do it yourself. But I will ask the chief pilot of Swiss Rockets to begin. Please, Vladimir.
Thank you so much for your introduction, Nenad. Hello, everybody. I'm very glad to be today here with you in Belgrade. I'm Vladimir Smiljanović. I'm a medicinal chemist by background, founder and chairman and CEO of the company Swiss Rockets, Swiss incubator and accelerator of biotech companies.
So I will continue. It's my greatest pleasure to be here with you. So I'm not coming from Serbia, but I feel like Serbian. Actually, I have Russian background, but sometimes I hear that Russians are coming from Serbia. So I'm Nadezhda Gracheva, so a scientist by background. I'm trained in radiochemistry, radiopharmacy, and I did my PhD in Switzerland in the field of nuclear medicine and specifically developing different radioisotopes for application in therapy. And I joined Swiss Rockets around 5 years ago, so I'm responsible for operations of different projects in nuclear medicine.
See, this is what I was talking about. Nadia is too modest. Please continue.
Great. Then what can I say more? So in the year 2019, me and of course my group where I was doing my PhD, we got Marie Curie Award of European Congress of Nuclear Medicine for the discovery of novel radioisotope that can outperform everything what exists currently on the market. So we brought this technology afterwards to Swiss Rockets and we continue its co-development for the needs of patients and their lives.
Now that's better. Thank you very much. Richard, please.
Yeah, my name is Richard Baum, and I'm obviously in love with Serbia because nearly exactly 10 years ago I gave a presentation here in Belgrade about a similar topic we are discussing today, 2014. I'm what you call a physician scientist. So I'm taking care of patients every day of my life since more than 44 years now, actually, and mainly with cancer patients, so patients having very malignant diseases, dying from these diseases, and the topic of the session is a cure, and that's what I'm dreaming about, and that was my motivation to bring me into medicine. I actually started in the field of cardiology, and then in the 80s, 1984, I started what is called nuclear medicine. I don't like the word nuclear, and many Germans also do not like the word nuclear, so I call it radio molecular. That means that we are working with molecules which you inject into the bloodstream and which are finding the tumor, which is called diagnostics. And the machine to do that is called PET or PET-CT nowadays. And then we can take the same molecule and label it with what Nadia is producing, a radioisotope which irradiates the cancer cell and then can cause a cure. I'm serious, I'm speaking about cure, because the first applications, the first use of radioisotopes in medicine dates back already more than 80 years. So in 1941, there was a colleague, a physician, his name is Saul Hertz, who was the first to use radioactive iodine to treat thyroid diseases. And we know that with radioiodine, with this radioisotope, you can cure cancer, even if it is metastatic. So not only the primary tumor, but even if the disease is in your lung or in your lymph nodes, for example, one can just by giving 2 or 3 capsules of radioiodine, one can cure cancer. And that was a fascinating example, but of course it works only for thyroid cancer, and we were interested in the 80s with monoclonal antibodies, which were just invented at that time, to bring this principle to other cancers, like frequent cancers, colon cancer, lung cancer, breast cancer, and so on. And we were quite successful in targeting and defining the disease, but not so successful in treating. So there was not a single patient cured from this treatment. That's why later on we went to what we call peptides or small molecules and labeled them also with different radioisotope. And I was happy enough to give the first treatment in Germany in 1997 for a very rare disease, an orphan drug disease, which is called neuroendocrine tumor. You all know what is insulin, which is produced in the pancreas. And these tumors can also grow and spread in the body. And these neuroendocrine tumors, and one very good example is the inventor of the iPhone, Steve Jobs, who had this disease and unfortunately died of this disease. And later on in 2013, we went to a very frequent disease, prostate cancer, which can also be treated and very rarely cured. So we are working to improve this, and you will certainly tell a little bit about that. And Elsin, what are ways to improve this radioligand treatment, how we call it, for prostate cancer? And then over the last 5 years, we were expanding to many different indications with a molecule called FAP. And this molecule targets all different type of cancers, but there is still a long way to go. So I was 22 years chairman of a department in a quite small city in the eastern part of Germany, and officially I'm now retired, but I'm still working 14 hours a day, taking care of patients in a city called Wiesbaden near Frankfurt. And I'm still enthusiastic about this idea of making a cure with radiation and molecules, with radiomolecular medicine.
Amazing. Thank you. Elsin.
I'm Elchin Zan. I'm a nuclear medicine physician, currently lead the nuclear medicine department at the Cleveland Clinic, and I'm a neuroradiologist as well. It's an honor to be here. More than being invited, the true honor lies because it's the country of Nikola Tesla, right? We owe him so much. As a neuroradiologist, I use MRI every day. And right now, with all the electromagnetic fields, all the research going in, it's really fantastic seeing that Bio4 has a mission to cure diseases. And once we cure diseases, hopefully your mission will be even further, more critically loaded with preventing diseases. So I'm passionate in providing better, perpetually improved patient care to my patients, and that's why I'm here. And thank you so much for having us once again. But that was the history, what Richard has given us, right? The future is how we can cure together, but at the same time, how we can screen and prevent so that wellness and health is shared all over the world, irrespective of where you're coming from. Thanks for having us.
Thank you.
I'm Vera Artiko. I'm a nuclear medicine physician, subspecialist in oncology. I have been head of the Center of Nuclear Medicine Clinical Center of Serbia for 10 years, and last 4 years, president of Nuclear Medicine Society of Serbia. Serbia has a very long tradition of nuclear medicine, almost 70 years, and very long tradition in radionuclide therapy and diagnostics, which, as Professor Baum said, began with application of radioiodine. So afterwards, there were some attempts, as I can recall memory, to use phosphor, I think, in hematological diseases, our colleagues from Medical Military Academy. And I think after 2004, we began treating of neuroendocrine tumors together with our colleagues in Vinci Institute. After that, our colleagues in Kragujevac continued that therapy and improved with the application of lutetium radiopharmaceuticals. And they also now apply those radiopharmaceuticals in treating of prostate carcinoma. In nuclear medicine now is improving very much. We obtain a lot of new equipment through IAEA projects mainly. With the emphasis on oncology, and also thanks to our Ministry of Health, we obtained a lot of new equipment, new PET-CT systems, new SPECT-CT systems, which will also improve diagnostics and not only therapy in oncology. In our institute, we are currently have under construction our GMP laboratory for labeling of gallium radiopharmaceuticals and lutetium radiopharmaceuticals, and I hope it will be finished until March.
Okay,
thank you.
Thank you. We've got a little bit of history, but I'd like to ask Vladimir, as a person who kind of develops and accelerates development of different kinds of drugs, why is radiotherapy so special? Why is this class of medicine so special and so hot topic in the world right now?
We have heard actually today George Yankopoulos, the pioneer of biotechnologies, about his story about antibodies. So you have molecules that can go specifically to tumor cells. We actually also, our company Tokyo developed, as many other companies, targeted cancer treatments, small molecules that can go specifically to cancer cells, to target them and to exhaust them or to kill them directly. But the new era is beginning with radioligand therapies, where you have molecules that can go specifically to cancer cells and to irradiate them. So you have, we call them torpedo molecules. You can inject them into a vein. They navigate through a bloodstream to the surface of cancer cells, where they are recognizing surface receptor proteins. And then, I like to simplify to say, then the radioisotope is acting like a molecular nuclear bomb, irradiating those cancer cells. And it has been shown, first time I saw that in the hospital by Professor Baum. I saw also metastatic patients where a significant amount of tumors was deleted or patients even cured despite metastasis, because those radioisotopes were able to break the DNA of the cancer cells, but also in many patients to activate the immune system, which then triggered the further curative treatment of those patients. So that's special. That's Novartis. We have Novartis colleagues today here with us. They did a very important breakthrough with their drug Pluvicto. Lutetium PSMA is the molecule for metastatic prostate cancer patients. This drug is prolonging the life of the patients with metastatic prostate cancer. But now new molecules are coming where people and scientists and companies are trying to improve the treatment of many different cancer indications while having more maybe promising torpedo molecules or radioligand therapies with new radioisotopes, with new targets, and combining that with different cancer treatments. That's where we see the future. That's where we see also curative treatments more and more in the near future, not in 5 years, not in 10 years. We see that also with our drug. We have the next generation Pluvicto, where we are using, instead of lutetium-177 radioisotopes that Novartis uses, we use Nadja's radioisotope. She was the first that developed that. Richard Bound, the first who tested in humans. The radioisotope is called terbium-161. So we see that we can bring more radiation into tumors, and that's where our company is focusing on.
If you allow me to add and to answer your question, what is so special? One point, you made it very nicely, Vladimir, is that we have molecules which attach specifically to cancer cells and not to normal cells. And the second, and I think probably more important, is that if you irradiate a cancer cell, you can really kill, you cut the DNA, so the cell dies. And a dead cell cannot develop resistance. All other therapies, if you look at chemotherapy, immunotherapy, and so on, they are all prone to the fact that the cells are very, very clever. So over billions of years, the cells learn to defend against everything attacking them, right? And then they can, what we call, escape. So if you treat a tumor with chemotherapy, initially, you kill some cells, but some survive. They become resistant. And the same is true for many, many immunotherapies, which are, by the way, very expensive. And that's another point one needs to discuss, is health economics. If a society can really afford these treatments, which were just presented but cost several $100,000, then it is very difficult, to have a cure for a frequent disease. But one of the specific features is that irradiation or radiation really can kill and cure. There is no resistance if you have high enough dose, of course, in the tumor.
Thank you. Nadja, you are, I believe, the expert to explain the rest of us. Those foreign or peculiar words like terbium, lutetium, gallium, iodine, and what's the difference and why do we have multiples of them and what's happening with that?
Sure. So, as was mentioned before, the word of radiopharmaceutical actually contains two main components. So it's radio and pharmaceutical. So for pharmaceutical, different companies we as well are developing biological molecules that will specifically go to the receptor in cancer cells. And it only will go to cancer cells because normal tissues will not express those receptors. Another important component is radio part, and here we talk about different radioisotopes. So there was a mentioning of iodine-131. This is the radioactive element that is being used for many, many years and successfully. So I believe this therapy will stay with us. However, the new generations of radioactive elements are coming. So one of them as was mentioned before, lutetium-177. This is so-called better minus emitter. So this element emits electrons constantly and irradiates your tumors while it's on the cancer cells. What we are doing, we're working on another radioisotope that is supposed to be more potent, and we know that it is more potent than lutetium-177. So what is the difference? It is also emitting constantly electrons over a long range, so it can irradiate different tumor masses successfully, but it has very, very short micrometer range irradiations that deposit a high amount of energy over this micrometer range. And this means that what we can do, we can not only treat tumor masses, but we can also address the main question of metastasis and micrometastasis. So that's what we are currently doing. We're developing molecules. Soon we go to phase 1 with one of our products. And we believe that this will be the future in that field, at least next level of what is currently existing.
Thank you. Thank you very much. Elsin, I'd like to ask you, since you originate from Europe and now work in US and probably have a more global view of what's happening, what do you use in your practice, and how do you see the development of the industry going forward?
The field is generalized as theranostics. Theranostics is a combination word, therapy and diagnostics. So I know everybody is familiar with chemotherapy, antibodies, small molecules, right? When you go to get your, when something is wrong in the human body, let's walk through this journey, right? You go to a doctor, they do a scan. It could be a CT, MR, or sometimes a PET scan. And then they take a biopsy of the tissue, and then they diagnose the patient. Then the treatment algorithm follows. What we do is theranostics. We image the patients with PET scans. We map the disease for the small molecule that we have to understand where it will go and how it will work. Because our therapies are famous for being highly impactful, but at the same time with really minimal toxicity. When you image the body, you can anticipate if there's a mass in the brain, right? We will be ready for treating the brain. Or if there is a mass in the liver, if there's a mass in the bone, is there a risk for fracture? So we are always focusing on safety first. If we do not feel treating that bone lesion with our therapies is not safe, we send the patient to radiation oncology for radiation before our treatment, or sometimes we treat them together. So the theranostics really brings the power of highly targeted therapies, delivering radiation, but at the same time highly safe therapies with minimized toxicity. This is our distinction from the others practicing theranostics so far for prostate cancer and neuroendocrine tumors. I work in the United States, so we follow FDA approvals. And these are the two indications, but right now it's a wide range. We have melanoma, we have lung cancer, we have colon cancer. There are so many different disease groups coming into the stage, but we have a big problem with drug development. Drug development takes years, clinical trial takes years. From batch to the patient for FDA approval, you're looking into probably 20 plus years of work. If we're talking about AI, right, we can do better, faster, still safer. So although I practice clinically prostate and neuroendocrine tumors, on a clinical trial research end, I do practice a wide range of diseases where I think we need to re-innovate how we perceive clinical trial design. In the US, it's very difficult because regulations are very stringent, which is great still, right? Safety first. Europe is very different as well. And I'm from Turkey. It's very different in Turkey, as well as Serbia being inside or close enough to the European Union. I think with the Bio4 initiative, it really brings this totally fragmented scientific discovery and then clinical applications in the patients from multiple fragmented regions to a holistic center where the drug discoverers, chemists, biologists, pathologists, physicians are all working together. And although a similar process exists in the US, namely NIH, it's still multiple layer of bureaucracy is really becoming one of the bottlenecks in developing drugs that are translated into the patients in a timely fashion. So in that sense, I would love to practice clinical trial design that is augmented, accompanied by AI, to make sure that we're not wasting time by the human mind-driven boxes, as we heard this morning. Human mind really thrives on creativity, because when you're creative, there's no box. And I think the Bio4 doesn't seem to have a box, really, will be full of super smart people.
Thank you. Richard, please walk us through the patient outcomes as an innovator in implementing those therapies and first in humans and doing all of those things in different illnesses, different stages of development of the illness. What are you seeing happening? You mentioned cures. You mentioned really true cures. So what could patients expect from those kinds of therapies?
Let me ask you, what would you expect if you have, let's say, a lung cancer or prostate cancer or whatsoever? What is your expectation?
Nothing less than forever cured.
Exactly. So you want to be free of the disease, you want to get a cure, but, and this is very important, with a good quality of life.
True.
Right? So the patients coming to me are mostly in very advanced stages of the disease, with metastases in the bone, in the liver, in the lung, in lymph nodes, and so on, and suffering from pain. This is one of the, unfortunately, most frequently observed symptoms, leading also to go to a doctor, is pain, right? You have something which is wrong in your body, you ignore it for some time, but then you can't ignore it because it causes a lot of pain, or you lose weight suddenly, right? You're eating normal and you are starting losing weight, and you don't know why the hell I'm losing weight, and you feel bad. Okay, so cancer is a disease which is very much impacting your life quality, as pain or as vomiting, losing weight, and so on and so on. And then one have to ask first, what is a diagnosis? So you have a symptom, you feel something, but you want to know why you have this symptom. And then usually, until now, you know, you start with what we call a physical examination. So the doctor palpates your liver, is auscultating your lung, and so on. Then you go to X-ray, then you go to ultrasound, you go maybe to MRI, if you have it. And if you get an appointment, right? For example, in Germany, an appointment for MRI is now up to 3 months. Wow. So if you have a breast cancer, do you want to wait 3 months to get a diagnosis? Or in United Kingdom, right? Also not anymore in Europe. You have to wait half a year or 9 months or so. So these are practical things you have to consider. And our aim with theranostics, which is part of personalized medicine. So this is what every patient, and you and me also want, is that the doctor specifically looks at your disease, not at lung cancer. Everybody is treated the same, right? But individually, regarding your age, you know, regarding your family situation, your professional situation, your working situation, and so on. So this is a whole health socio-economic system, right? It's not only a diagnosis, breast cancer, right? It's affecting your family, your work, your life, your quality of life, and so on. And this is where AI, and I hate the word AI, to be very open. I think it's the wrong word. It's not artificial, right? And it's not an own intelligence. I call it IBM, not like the good old blue company, but intelligence-based medicine, right? So we should use intelligence to make a new approach to medicine. And I think IBM, or some call it computational medicine, probably too complicated for many to remember, but it's really statistics, right? It's numbers. And there was so much talk this morning, right? But very little numbers, right? What we need are data to get into the system, right? And this is where our approach with Theranostic starts, to get data in a smart way from the patients. So in future, you use your watch, right? You're walking around, and when you are going to the doctor already knows your weight, your age, your blood pressure, your pulse, your temperature, and probably even your symptoms, because your iWatch can already tell, you know, where is your pain, how intense is your pain, and so on. Though there are very, very smart ways to get information flow from the patient to the doctor, and then to personalize this information. And as Elsen absolutely correctly mentioned, then you have to find a smart way to make a fast diagnosis, not X-ray, MRI, ultrasound, and after 3 weeks, you don't know, you know, if the lump in your breast is a breast cancer, if it is an aggressive breast cancer you will die from, or it is like in many older women, just a breast cancer you can observe or give a pill and stop hormonal production and get the same outcome. So this is, I think, what we really need to reach with intelligence-based medicine or AI, you know, to make a personal approach to your treatment and to get the information flow. And on the other way, if you are treating diseases, you have to get the data to improve the treatment. So nowadays, there are thousands of treatments done every day. Nobody is looking at the follow-up data. Nobody is, you know, analyzing this data in clinical trials. Not only for new drugs, for routine treatments. We just need to analyze and get data. This is AI, right? It's really, you know, a new way of thinking and getting data in for the improvement of the treatment and for this personalized approach, which is very easy to remember. It is the right treatment, the correct treatment, for the right patient, for you or for you, okay, at the right time, so when to start the treatment, and at the right dose or medicine, right? Using terbium, for example, at the European Congress, where I'm just coming from. I was there yesterday in Hamburg with 8,000 people now visiting Nuclear Medicine Congress in Europe. Some years ago, it were 2,000 or 3,000, right? Annoying and saying, oh, nuclear medicine is dying. Now everybody is very excited. Theranostic is blooming because of this new approach and the fact that patients really have benefit from these treatments. It's not only talking, right? You get improvement of pain, you get better quality of life, and this is not just I'm saying that, that is shown by clinical prospective randomized trials, and this is where industry had a great impact, you know, on bringing these new therapies in.
Thank you. We are all probably eager to hear what's the situation in Serbia and how important is international cooperation with all those smart people and many more for Serbian healthcare system, for Serbian patients, and for Serbian practice of radio.
Thank you for the question. The situation in Serbia is follows. we have a lot of radiotherapy units. It means units for treating of carcinomas in Oncology Institute of Vojvodina, then in Zrenjanin, I think, Institute of Oncology and Radiology in Belgrade. It is under construction in our department, University Clinical Center. We have a University Clinical Center in Kragujevac, University Clinical Center in Niš, and Chigota Zlatibor. It is too much. Even in one hospital in Chuprija, we also have therapy unit. Most of that units have been dealing for a lot of decades with thyroid carcinoma as we began. But last 20 years, we have been doing therapy of neuroendocrine tumors and prostate carcinomas as well. What can be a potential problem for us, and is a problem, that we cannot provide radiopharmaceuticals as new and innovative as we would like. We have very strict regulations here, so we must provide all radiopharmaceuticals acknowledged by our Agency for Drugs and Medical Devices, which is okay. But radiopharmaceuticals, as Professor Baum said, are developing very fast. So there is no time to wait for radiopharmaceutical to be registered, because when it is registered and we use it, it has already been out of use in most of the European countries. So we need faster application of radiopharmaceuticals in our daily practice, and with Minister of Health we are doing that, trying to change legislation, I think. And also the problem could be the cost of the therapy, but still it is the second thing.
Thank you very much. Vladimir, before opening up to the questions from the audience, could you please walk us through a business of developing and running this drug development as a business? You do have a company that's particularly devoted to radiopharmaceuticals or radioligand therapies. And how does it look from the global perspective? And if you're an entrepreneur wanting to develop something like this, how does it look like?
Thank you for that question. So the pioneering work in regard of radioligand therapies has been done actually in two centers, one in Basel, in Switzerland, and in Bad Berka, the clinical part, Professor Baum. But then biotech companies and entrepreneurs behind in Switzerland and in US, they did an important clinical development part and development part, and those companies were sold to big pharma companies, to Novartis especially, and then later to other companies. And Novartis was the first big pharma company making a true innovation on the market, bringing those molecules to the market to cancer patients, like products Pluvicto for metastatic prostate cancer, Lutathera for pancreatic neuroendocrine tumors, and now other big pharma companies and biotech companies, they are developing next generation radiopharmaceuticals also for prostate cancer, neuroendocrine tumors in general, but also for other cancers. Our company, Torpedo Pharmaceuticals, the daughter company of Swiss Rockets, is actually utilizing, as I said before, the new radioisotope, and that's how we are trying to differentiate us. Also in prostate cancer, we go into those patients where Pluvicto doesn't work anymore. This drug of Novartis is prolonging the patient's life for several months, but afterwards patients still die, the majority. What we are doing on one side is trying to prolong the life of those patients who failed on Pluvicto by using another radioisotope, Terbium-161, or we go into early stage patients. And going into early stage patients, that's where I believe with that radioisotope we can cure patients. And that's something where I'm strongly a believer. And according to the data that we have so far, and what we are doing as well in regard of business is we are probably the first working on the specific radio-sensitizing molecules. So what we want is first to exhaust the cancer cells, and when we put them actually into exhaustion state, then we want to send the torpedo molecules to actually kill billions of cancer cells more efficiently than using radioligand monotherapies. So as an entrepreneur, we are trying always how to differentiate, how to overcome the challenges and problems that first-generation radioligand therapies have, and that's what entrepreneurs can do. Swiss rockets, the name is rocket, so we are trying always to do that with a rocket tempo. And then when we show that this works, then we partner usually with big pharma companies. Novartis is our neighbor in Switzerland. First we try to compete, and when they see that we are very competitive, then usually this ends in a fruitful collaboration.
Thank you very much. I have a lot more questions, but it's turning red on the screen, so I'd like to open up for the questions from the audience. We have a couple of minutes. Just please, quick questions and quick answers so we can accommodate as many as possible. I see the hand over there was the first one, I believe, but not really sure if do we have the mic, or shall we share the mic? Use this one for the audience. And
Hello, thank you for this wonderful talk. My name is Boban Stanovic, and I'm from Great Ormond Street Hospital for Children, UK, London, cell and gene therapy group. And before that, I led clinical trial at King's College, London, all in cancer, mostly leukemia. Before that, I did 3 postdocs in Japan, in Kyoto. So I have I would say, good experience in cancer. And I want to defend here our delivery system. We use lenti and retro to deliver. You never mentioned here. I would like to know your experience or something, or where is the future? Small molecules or lenti, retro, as we can see from Professor Koppola's antibody. But we have quite success with lenti and retro. So
who's the question for?
Any.
Okay, who'd like to take this?
Can you please elaborate on lenti-retro? You mean the viral?
Yeah, lentivirus to deliver gene to the cancer. Yeah.
CAR-T therapy, right?
Yes. Yes.
Definitely. Also CAR-T therapy. Novartis is the pioneer there. We have spectacular data in CAR-T therapy. What does it mean? You take from a cancer patient blood, you take the immune cell, you go to the laboratory, and with the lentivirus, you actually equip your own immune cell how to molecularly recognize better the cancer cell. You replicate that modified immune cell and you bring it back to the patient. And we saw there revolutionary results, revolutionary data. Novartis is the pioneering big pharma company behind. And what we are also trying to do is to combine radioligand therapies, antibodies, small molecules with immune therapies, like CAR-T therapies or checkpoint inhibitor antibodies. So definitely those therapies can do big success. But CAR-T therapy, for example, there are no very good examples where this therapy can cure patients in solid tumors. In leukemia and liquid tumors, yes. But in solid tumors, we don't have success like in liquid tumors. So research groups all over the world are improving the research, trying also to improve CAR-T therapies for solid tumors. And me personally, as a big doctor, Richard Baum, there as well, for me personally, the combination of those particular technologies and therapies can help us also to cure patients, and that's the biggest hope that I see so far.
And for the one comment, CAR-T cells are probably going to be improved massively, but scalability is a big issue. You need to extract, expand, give it back to the patient. But our therapies are done outpatient. A prostate cancer therapy drug takes only 15 minutes to infuse, once we know the patient has the receptor expression. And so on. I think scalability and access will be very important. Right now, as we heard, that's great. There are 7 centers in Serbia who can deliver radiopharmaceutical therapies, but how many CAR T cell centers?
0.
So, and
we use patient cells we modify them, so it is not CAR T. It's a little bit different.
I encourage you to continue this conversation after the panel and just have one more question for the. And then the other questions will unfortunately need to be addressed after the panel, but just one more quick question, Jens.
Thank you very much. Inspirational talk. I have a question about combination of immune therapy and radiation therapy. If you tag your radioisotope with the molecule, for example, protein, and you design that protein to interact with the cancer cell, how a radioisotope affects structure and dynamics of that protein, of that molecule. So my question is, for example, you use Dr. David Baker's Rosetta Fold-based tools to design ideal protein that will interact with your with a protein from your target cell, and then you attach a radioisotope to that protein. How that radioisotope affects that? What if simply that protein cannot interact with your desired target?
Maybe Nadia can answer that best, but it's very well studied, right?
Yes, so basically, before we give the therapy, you always ensure that your molecule will go specifically to cancer cells and that there will be an interaction between your radiopharmaceuticals. So that's what Richard would mention before, theranostics. So we do first specifically diagnostics. You take the same biological molecule and you attach very short-lived radioisotope that you can use to do PET-CT scans. So you can visualize and you can predict where your therapy goes. After that, you use therapeutic radionuclides and then basically also visualize where it goes.
I understand that. The question is, you have a natural biological molecule, and but that molecule didn't evolve with, in the presence of radio.
Yeah, but that's very well, excuse me, that's very well studied since 40 years, right? We know about radiolysis, about radionuclide purity and so on, so labeling efficiency. And there have been, meanwhile, hundreds, thousands, if not millions of patients treated with this radiopharmaceutical. So we're very well known from praxis. It is a problem, specifically for alpha emitters, for example, for radioisotopes with a very big power, destructing a protein or so. But that's a well-known problem, and I think it's for practical purposes solved.
Exactly, and sorry, just to add here, so basically before you give a radiopharmaceutical, you ensure that it's stable, you ensure that what you inject is actually only your radio-labeled compound. So with years we learned how to make the right formulation, and it may differ for different isotopes, but generally speaking, we learned how to do that. Yeah.
And as it should be for such a hot topic, the discussion will continue. Thank you very much.